Hello friends, welcome back to my channel BioFrick. In today's lecture video, I'll be talking about topoisomerases. Now, topoisomerases are the ones that relieve the strain over the DNA duplex as DNA helicase unwinds the DNA. So basically, as it unwinds the DNA, there will be replication fork that will be formed, but upstream and the downstream region of DNA duplex will be supercoiling them. So that has to be relieved by something, and topoisomerases do the job. Now, when we talk about prokaryotes like E. coli, DNA gyrase is type 2 topoisomerase that does the job, while in eukaryotes we have topo 1b. Now, how do they actually function? Okay, that's really very interesting process, right? So they have nuclease activity. Okay, they are nucleases. Basically, they'll cut the DNA somewhere, right? So they'll break the phosphodiester bonds. Okay, and breaking and making, it's basically the reversible process there. Now, under topoisomerases, we have type 1 and type 2 topoisomerases. Now, type 1 topoisomerase cleaves one strand of the DNA and the other strand of the DNA passes through that single strand DNA cut. Okay, so you can have a look at that image there, or the drawing there, wherein I have drawn the double stranded DNA. One is red strand, other is blue strand. Red strand is nicked. Okay, there is a single strand DNA nick that is formed by topoisomerase, and the blue strand will pass through that nick and you will get the relaxed DNA formed there. Okay, so there is negative supercoiling that is occurring. While when we talk about type 2 topoisomerases, it's dealing with the double strand cut. Okay, so when there is supercoiling, okay, the double stranded DNA, when it's coiled like that, okay, as you can see in the image there or the drawing there, okay, it has to be cut on the double strand. So one whole double stranded DNA is cut there, and the other double strand will duplex will pass through that, and there'll be relaxation, negative supercoiling of that particular portion. Now, another difference here is while naming these topoisomerases. So whenever you see the odd Roman numbers after the topoisomerase, like topo 1, topo 5, okay, these are under type 1, while type 2 have even Roman numbers after their names. Now, type 1 also doesn't depend on ATP hydrolysis except for DNA gyrase, not DNA gyrase, sorry, reverse gyrase. Now, reverse gyrase requires ATP, which is exception under type 1 topoisomerases. Otherwise, type 1 topoisomerases do not require ATP. Reverse gyrase is the one that will introduce positive supercoil. Okay, and type 2 topoisomerases, they utilize ATP for hydrolysis, and they are not dependent on any external energy source. So let's look into details of type 1 topoisomerases. Now, in order to understand different types of type 1 topoisomerases, we need to understand that topoisomerases have tyrosine residue, which is nucleophilic. Now, this tyrosine residue that is nucleophilic, it promotes the strand cleavage. How it will form, this tyrosyl oxygen which is there, it will attack that particular strand, it will break the phosphodiester bond, and it will form a transient phosphotyrosine bond, transient covalent phosphotyrosine bond, and it is later rejoined by transesterification, which is reverse of the previous process wherein there is breakage, and now it is rejoined by transesterification. So basically, type 1 topoisomerases, they operate by forming transient phosphotyrosine covalent bonds, okay, with one end of nicked DNA. So under type 1, we have type 1a and type 1b topoisomerases. Now, how to differentiate between two? Okay, so tyrosine will covalently bind with 5' phosphate in type 1a, and in 3' it will covalently bind to 3' phosphate in type 1b. How to understand this? Okay, look at the drawing down there. Okay, so here you can see that there is type 1a, whole topoisomerase is coming there along with tyrosine residue, that is tyr I have written there. And now it is forming transient covalent bond with phosphate, right? Phosphate, but it's 5 dash phosphate. If you look at that red strand, it is in direction 3 dash to 5 dash, so 5 dash phosphate is interacting with tyrosine. But when we talk about type 1b, it is interacting with 5 dash to 3 dash phosphate, and that 3 blue strand, right? So phosphate is interacting with tyrosine of type 1b topoisomerase. Another major difference is that type 1a passes the single strand DNA through the transient break that is formed, right? The previous one, whatever we spoke about, the topoisomerases type 1, I said that it passes through the nick that is formed. Okay, one single strand passes through the nick. That is true for type 1a, but in type 1b they have a different mechanism, okay, in order to introduce negative supercoil. How that is done is that the downstream region, or it might be, upstream, but usually the downstream region, it rotates with respect to the other duplex. So nick one DNA strand, okay, it will nick, the topoisomerase type 1b will nick one DNA strand, and duplex end rotates with respect to the other. Okay, so as it rotates, what's happening is basically it is revealing the strand, and one coil is lesser, and one by one it will introduce the negative supercoiling. So type 1a has three different classes under it. So topo 1a, topo 3, reverse gyrase. Okay, reverse gyrase introduces positive supercoil. It's true, and it's found in eubacteria and archaea, while topo 1a and topo 3 introduce negative supercoil in eubacteria and eukaryotes respectively. Talking about type 2 topoisomerases, right? We spoke about the two different types under type 1, which has type 1a, type 1b. Now type 2 as well has type 2a and type 2b. Now before that, as I said, there'll be double strand break, right? So this double strand break uh, is basically the G segment. Okay, so the strand that has double strand break on its helix, okay, is G segment, while the other one, the other double strand that passes through this particular break, that particular duplex is said to be G segment. So T segment, sorry. So T segment passes through G segment. Under type 2 topoisomerases, we have type 2A and type 2B. Type 2 has topo 2 in eukaryotes, topo 4 in bacteria, DNA gyrase in bacteria and archaea. Okay, this is the one that is important in DNA replication in E. coli as well. We spoke about DNA gyrase there. And type 2b has topo 4 that's seen in plants, sorry, topo 6 that's seen in plants, and SPO 2 in east. So that was it, guys. Hope the flowcharts were helpful. You guys can take screenshot of it or draw it by yourself and understand the concept. Thank you.